the seaweeds, but extensive research being made now on microalgae. Designing new products, that's also a way of looking for new approaches and also for a better understanding of existing products. So we will also talk about protein hydrolysates. Again, protein hydrolysates, yes, but looking to the detailed information about what individual amino acids can do, how the molecular mass of the peptides matter. So all these questions will take us for the afternoon, but first of all, I would like to introduce our keynote speaker, Professor Uwe Conrad, who is Professor of Plant Biochemistry and Molecular Biology at Aachen University and a co-founder of AgPrime and GmbH company, and a scientific advisor to agricultural and biotech companies. In particular, is passionate pioneer and acknowledged world leader in the research of defense priming. The term describes the enhanced capacity of immunized plants to mobilize defense, resulting in earlier, faster, and more robust activation of defense, which often leads to immunity. Uwe's research also addresses various ag-biotech and bioeconomy aspects for translating concepts discovered in model systems to agriculture. They include tools for spotting, priming-inducing compounds, defense pathway engineering, and leaf-sticking gels and capsules for the slow release of active ingredients. Maybe it will look a bit surprising that Uwe Conrad will talk a lot about biocontrol and more than biostimulation, but it was a decision by the scientific committee to invite Uwe Conrad because he's one of the main pioneers of this concept of priming and of the application of this concept of priming that comes indeed from the biotic stress field, but we know how relevant this is also for abiotic stress, and I think it's important to listen to him. Uwe, you have the floor.
Yeah, thank you. Thank you so much, Patrick, for these nice words. I hope it wasn't over the top. And I also would like to appreciate being invited, also Patrick and all the others in the committee for inviting me here. Yes, you're right, I'm kind of a other planet here, but I very much enjoyed the talks, and I think maybe we can, in the biocontrol and in the biostimulants area, synergistically drive the field. Yeah, I would like to get started, ladies and gentlemen, with introducing you to an experiment that Frank Ross did in 1961. Frank inoculated a tobacco plant with tobacco mosaic virus, and to no one's surprise, after a couple of days, he has seen aquatic lesions. And then he did the same experiment with a tobacco plant of the same batch of seeds that he had inoculated 1 week before with tobacco mosaic virus. And as you can see over here, in the inoculated systemic leafers, as we say, there was hardly any lesion in comparison to the control. And the few lesions that popped up, they were substantially smaller than those that appeared upon primary attack. There also was broad spectrum disease resistance seen. So the resistance that developed here is active not only against the inducing TMV, but also against other viruses, against fungi, and against oomycetes. So it's broad spectrum disease resistance that he induced. And not much appreciated in the community, there is also enhanced resilience of that plant to heat, cold, and drought. Frank referred to this phenomenon as systemic acquired resistance, and this is actually what I'm going to talk about most of this afternoon. When you go back to the literature, the phenomenon is not, it's very old. You can get first reports from Julien Rey and Jean Bouverie from 1901, French botanists who described the phenomenon right after the beginning of the century, of the last century. And since then, Plant biologists wondered about what's going on in the systemic tissue, what makes the plant systemically resistant. Well, it was only in the early 1990s when the labs of Dan Classic and Jean-Pierre Metro and John Riles, when they reported as a summary that upon infection of a local leaf with a pathogen, systemic signaling gets induced, and this leads to the systemic accumulation throughout the plant of salicylic acid, a small phenolic molecule. Salicylic acid then causes the accumulation of antimicrobial so-called ZAR proteins, and many of these proteins are PR proteins, and many PR proteins have antimicrobial activity. PR2, for example, is a glucanase, PR3 is a chitinase. There are small chitinases over there and also glucanases over there. And then people thought, well, issue solved. This is how SAR works. The systemic accumulation of the antimicrobial proteins is responsible for the enhanced defense capacity of the plant and the resistance seen in the systemic tissue. Okay, this then leads to systemic immunity or systemic acquired resistance. Well, and then this whole concept was challenged again because it was especially one of the world leaders in PR protein research, that was Kees van Loon of Utrecht University, who wrote in a book that he published, or in a book chapter published in 2000, that the overexpression of PR genes in transgenic plants has shown that the overexpression of PR2 and PR3 that accumulate during ZAR do not confer resistance. And taking the evidence together, Kees stated that the contribution of PRs to SAR appears to be minor. And I can still hear his heart plugging because he was really for 30 years the pioneer of PR protein research. So the issue was open again. How does a plant express systemic acquired disease resistance? Well, I was a PhD student in my last year at the time, and I have done some experiment that I introduce you to in a minute. I had the sneaky feeling I can find an explanation for why a plant gets systemic resistant, but I didn't know anything about SAR. So what I did is I sent an application to Dan Classic to the United States, and fortunately enough, he invited me to be a postdoc in his lab. So I went there, learned everything about SAR as I could, and then I went back to Germany, and I followed up this series of experiments that I did before. And I was actually interested in how does a parsley cell culture perceive a pathogen signal and transduces into a cellular defense response. So when the paper about the important role of salicylic acid in SAR was published, I thought, well, salicylic acid maybe is a good inducer of defense responses in plants, and that's why it makes the plant resistant. So what I did is I used the parsley cell culture. I added salicylic acid at pretty high concentration and even for 22 hours. And what I've seen is not that much. This was long before global gene expression analysis. I had just 4 marker genes. As you can see over here, salicylic acid does not or only a little bit induce these defense genes that I measured over here. And then I used a very low dose of an elicitor, and because it's a very low dose, it also did not do much. And as you can see over here, this is the phytolexins. They get secreted. It's antimicrobial compounds that get secreted to the culture medium. Nothing happens over here. And then I did the key experiment. I pre-treated the parsley cells for 22 hours. I waited 22 hours, and then I added the elicitor, and the result was just amazing. As you can see over here, there was a huge expression of all those 4 defense-related genes, and there also was a huge secretion of antimicrobial phytolexins into the culture medium. So, to make a long story short, salicylic acid does not per se induce defense much. It rather, during 24 hours, switches the cells to a state that makes the cell sensitive to pathogen signal. And when it gets that signal, it boosts the defense response, and this, to my opinion, is then leading to systemic acquired disease resistance. I took the term defense priming from the mammalian field. It was known, and there's many examples. I just brought one from the Stewart group. it was known that if you pre-treat cells of the innate immune system, here the mouse L929, so this is sarcoma cells, if you pre-treat them with interferon and you then stimulate them later with Newcastle disease virus, you get much more interferon defensive compounds as you do it if the cells have not been pre-treated with interferon. So the same phenotypic phenomenon in the mammalian field, which was called defense priming, this is where I took the term. And actually Joe Kuch in the US, he also used the term a couple of times. Okay, so the child was born, and we thought when we translate this to intact plants, what is going on? And the speculation was upon pathogen attack, there is systemic signaling going on. Then there is systemic accumulation of salicylic acid throughout the plant. And today we know that N-hydroxypipergolic acid also is a signal that systemically accumulates and acts in concert with salicylic acid. This then activates genes encoding SAR proteins and pathogenesis-relating proteins, but they are not that important. Rather, what they do is they induce this state of defense priming, that state of the alert that needs time to develop. This turns the traffic light yellow, the plant is ready to go, and when it then comes to an infection, then there is systemic acquired disease resistance just because the plant cells are ready to boost their defense response. Okay, so the child was born. We announced this in 2 reviews that were highly cited since then, especially that one over here, something like 1,600 times. And that was followed by many other reviews, so we had quite some invitations to get the principle distributed in the community. What about mechanisms? I don't have the time to go in all the mechanisms we know about defense priming. I just want to give you a short summary of those that I find the most convincing. So we have shown in 2009, and this was work by Gerald Beckers in my lab, that in primed plant cells, in Arabidopsis thaliana in this case, there is accumulation of proteins with a role in signal transduction. And because there is more signal transduction proteins in primed plant cells, there is a better signal transduction upon stimulation, which then leads to the boosted defense response. In 2014, Gene Greenberg's group at the University of Chicago has shown that primed Arabidopsis cells also have more receptors for pathogen signals. So because there's more receptors for pathogen signals and more enzymes that transduce the signal, there is a boosted defense response eventually. And we have shown in 2011 that chromatin modifications provide a memory for systemic resistance in the plant stress response, and we have recently seen that this is also true for biostimulation. I want to guide you through that. So, and I know, of course, here in this audience, I'm not talking to 100% molecular biologists, so I try to give you the information that we need to understand what I want to say. So, you know probably that our DNA is not naked. Eukaryotic DNA is wrapped around proteins, the so-called nucleosomes, and they contain of histones. And the N-terminus of the histones can be modified. So DNA is wrapped around proteins, and those proteins, they can be modified. And the modification of these proteins has an information content. For example, and this is a busy slide now, don't get confused. Just check here. Lysine 4 can be methylated, mono-di-and-3-methylated. Sorry, histone H3. Lysine 4 can be mono-di-and-3-methylated. Let's just have a look at this one. This is a very important modification because from yeast to man to plants, it is known to be associated with gene activity. So we checked a defense gene. It's a transcription factor that is involved in plant defense, and we have seen no. We see with PTH, it's a synthetic salicylic acid analog, we do see that the treatment induces the modification, but the gene remains off. I had the sneaky feeling, hey, this is probably the memory of the plant. This mimics a defense, the defense. Then in the systemic tissue, the modification is written. It prepares the gene without activating the gene, and when it then comes to a secondary stimulation, the defense response, the gene expression is way stronger in the primed plant as compared to the non-primed plants. And then we did, to my opinion, the master experiment in this series of experiments. We did a local infection of Arabidopsis leaves with bacteria, Pseudomonas cannabina alasalensis, and we checked in the non-infected leaves for the modification. And you can see all those modifications at different defense genes were written. This is the memory of the plant. This is the memory of a plant to get ready for a secondary infection. And when we do the secondary infection, those three genes are way stronger expressed in primed plants as compared to plants that have not been primed before. We are talking here about the memory of a plant to an infection. It stays for about 2 weeks in Arabidopsis, longer in crops. Why is this interesting here in this audience? Zoya Avramova at the University of Lincoln, Nebraska, did a very interesting experiment that was just followed up our study. She had Arabidopsis plants. She took them off soil and put them on the bench on a paper towel for 2 hours, which is kind of a drought stress. Then she puts them back for 22 hours. Then she takes them off again for 2 hours, puts them back again and again and again and again. She checked the expression of two drought-responsive genes, as you can see over here. See here, watered, stress 1, gene gets activated. Recovery phase, and then second stress, expression of the gene way stronger. Same is true for this gene over here. But check it out here. This is watered, stress 1, faint response, modification written on the promoter. Stress 4, very strong response, even more modification. So the stress 1 induces this modification, it stays on the promoter, and it gets even higher with subsequent stresses. So she trained the plant for enhanced drought tolerance, for enhanced drought resilience. And again, the histone modification H3 lysine 4 trimethylation is the memory of the plant to the stress. True for biotic and abiotic stress. And to no one's surprise now, you can see here, after the stress 1, the plants do not feel well. After the drought stress in S4, where the plant has trained and has the memory, it looks more or less perfectly fine. So this brings me to application. You can see we, over the last 20 years, have had a journey with many strong industry partners, not all of them working with us in the priming field, but most of them do so. It all started with a collaboration with BASF many, many years ago. They phoned me, and this was when just the strobilurin fungicides, the headline, or pyraclostrobin was introduced to the market. Shortly before, they phoned me and they said, we've made observations with the compound in the field shortly before market introduction, which we do not understand. You can see this over here. So this is the interaction of tomato with cucumber mosaic virus, sorry. These are the control plants. This one has been inoculated with virus. These ones look better than even the control, and they have been treated with pyraclostrobin, a fungicide, and then inoculated with virus. They perform even better than the control. Look this up over here. Corn plants in the field, untreated, treated with fungicide. The field experienced drought. Those guys over here that were treated look perfectly fine. Also the crops look way better than those that come from this field over here. So this compound, a fungicide, inhibits non-target organisms and provides drought resilience to the plant. And they didn't understand that. The only explanation is something must come from the plant. Something must go on in the plant. And so we did a very simple experiment to clarify this. The simplest experiments are almost the best. We just remember in the beginning, Frank Ross, tobacco. We just sprayed tobacco with pyraclostrobin at different concentrations. And then after 1 day, we inoculated with tobacco mosaic virus. This is the control, and you can see there's enhanced virus resistance in tobacco after treatment with pyraclostrobin fungicide. And then we checked on a molecular basis whether this induced resistance is due to priming, and you can see that in the pre-treated plants, which have then been infected with virus, there is earlier accumulation of antimicrobial PR proteins as compared to the control. So the plant is clearly primed by pyraclostrobin. And this was a top seller especially for the headline in the US. Farmers used it more as a booster than as a fungicide. So this is needed. Chemical industry wants priming. So how to identify priming active compounds? I've been asked this often, and then I said, well, of course we have the system, we have the partial cell culture. So what we do today is, when our partners come to us and they want to know whether their compound is a priming compound, we usually use microtiter plates. We have salicylic acid as a positive control. We have their compounds. We pre-treat for 24 hours to induce the priming or not. And then we stimulate with the elicitor again, and if there is positive hits, and these are 3, we do see the fluorescence, very easy, very reliable assay, and not very much dependent on where the compound comes from, so it's really very reliable. So we use this, for example, to find, you can see here, salicylic acid as the positive control, boost the defense response in the primed condition, not boosted in the unprimed condition. Pyraclostrobin does the same. Clear evidence, pyraclostrobin is a priming inducing fungicide. Sulforaphan we found, it's an antioxidant, an isothiocyanate, also primes plants for enhanced defense. It's very, very interesting because this is a compound that is a histone deacetylase. So here is the epigenetic memory again. We published this. We have shown that sulforaphan modifies the histone, unpacks the chromatin, and primes defense in Arabidopsis. For drought, for inducing, training the plants for drought tolerance, training them for induced immunity, 2 events are important. I told you already the modification of the histones. The other one is the RNA polymerase, that's the enzyme that reads a gene, must be phosphorylated. We've been asked by many guys, many companies, especially the Bayer company, can you do a plant to identify a priming compound that works via the epigenome? Because they are expecting these are probably the most powerful compounds to induce priming. And I said, yeah, we have it already. So what we did is we used a mutant of Arabidopsis, which has a defect in the phosphatase. So the RNA polymerase always wants to go, always wants to translate genes. So in this plant, we have only to induce the epigenetic modification to get the primed expression of the gene. So that's what we did. You can see this over here. This is the control, so it's the wild type. We used the promoter of a defense gene. We used GFP, which gives the fluorescence, to read out the response. And you can see here, this is the control. The mutants are a little leaky. Also, the RNA polymerase wants to transcribe, so it does a little bit. Here is PTH, the salicylic acid analog. Here is a compound of Bayer, but this is the wild type. Here's the real thing. This is here our reporter plane. This is the control. Here treated with the salicylic acid derivative. On here, the Bayer compound. So an easy spot. Fortunately, we patented it before, and Bayer now bought a patent a couple of years before, and now they use it heavily, that I can say. They allowed me that. They heavily use it in their screens for novel compounds, and this one over here is actually in the pipeline to become a marketed product. The problem with those compounds is many of them are washed off. Whenever you use a compound, especially when it's a hydrophilic one, they get washed off. So we thought about an approach, how can we keep compounds, maybe some that are under pressure, on the plant? And we developed microgels. They are about 200 nanometers to 10 micrometers. You can actually include here whatever compound you want to. Hydrophilic is better than hydrophilic. We attach proteins that have a hydrophobic residue that sticks to the waxy cuticle to the wax layer on the plant, so it sticks to the plant, the gel is there, the compound is in there, and now we've made it tunable. So, very good, for example, for fungicides or copper or something like that. So we have the compound in the gel, it is dry, and now it gets wet. Water is coming. The compound gets released because the gel is swelling. Compound release, release, release. It will fight a pathogen when it is wet or humid. Or so. This is especially true, as you know, for fungus and for oomycetes. And then it collapses again when you take off the water. So this is a reservoir for antimicrobial compounds, especially when you want to use not as much as you usually would use. We've seen this with grapevine, the copper effectiveness. This is not really correct. Copper effectiveness to downy mildew is not 5-fold enhanced. We needed only 1/5 of the amount of the copper when we use those gels to get the same effect as we use 5 times more of the copper chloride. And we recently did even another step. Patrick already mentioned it. We, 2 years ago, funded AgPrime, a company that was funded by myself and a couple of my group leaders and postdocs. We want to help further translate priming to the field. So what we are doing is, we are now 10 people. We are located north of Aachen. This is Aix-la-Chapelle or Charlemagne, 20 kilometers north of Aachen in the ITS. It's a center. Some customers want to be disclosed, others don't want to do so. I just talked to guys from Yara, so greetings. They also allowed me to disclose their ID over here. So we have a couple of selected customers who are interested in priming. What we do is basically we use extracts that we got from our customers, fractions, molecules, or organisms. We check formulations, develop formulations with our partners. We look for synergy with conventional products. Also with synthetic fungicides or pesticides in general, we check for plant health and abiotic stress resilience, identify health, abiotic stress resilience promoting activities. We do mode of action analysis, evaluate active ingredient performance, etc. We also develop screening tools depending on the needs of the customers, and we also have identified our own priming compounds, agents, and beneficial rhizobacteria. We do this on different levels, from cell cultures to check for priming capacity, over leaf disc, detach to intact plants to check and see whether there's priming activity. So when you have a molecule, an extract, a fraction, a mixture, if you want to know whether your compound isn't using defense priming in plants, because it's always a selling point, just talk to us. You will find us on the internet. With that, just in time, I would like to thank all those who supported our research over the last decades, and I very much thank you for showing up and listening to me. Thank you.
Thank you very much for driving us through this journey in the wonderful land of priming, which is definitely exciting. And from molecule discovery to molecule delivery, which was a nice journey. We have time for questions. Patrick, yes.
Thank you, Uwe. Great talk.
Thank you.
You noted that things like strobilurins developed as fungicides have abiotic stress, drought stress tolerance. You mentioned in some of the original work that crossover between abiotic and priming occurred. So 2 related questions. How specific is a primer to a range of different biotic and abiotic stresses, number 1? And since all that a primer does is initiate a constitutive gene, it was a capacity the plant always had. So if the plant always had that capacity, why was it not always turned on?
The second question I didn't get, Patrick.
So what the priming does is prepare the plant for a response to a future stress.
Right.
Okay? Why, and it lasts 2 weeks or 3 weeks, why, if the plant already had that capacity, does it not express it at all times?
Okay. So the second question I answer first. It's just because priming also costs energy. You have a little bit of a fitness penalty. So when we use a plant in sterile condition and we prime it, we can see a little bit of a fitness penalty, definitely. So it's visible. And I think that the plant is not primed all the time just because of that. It's different in the field. In the field, you have a mixture of primed, induced, and non-responsive planes. There's a refractory phase. And then you also can ask the question, so why aren't they primed all the time? Actually, many of them are, but what you do with a priming agent, you put them all on the same level. So you do a reset. Primed, let's go, right? So in the answer to your first question, it's broad spectrum. So priming in systemic acquired disease resistance is active against all biotroph and hemabiotrophs pathogens that we tested, not against necrotrophs, but non-necrotroph, at least in our lab. And it is also effective against many abiotic stresses that we didn't follow up that much. Yeah, we are more in the control, in the biocontrol, but it's really broad spectrum and it includes also the abiotic stress. The reason is just we hardly look for abiotic stress. And guys in the abiotic stress field, they hardly look for pathogens. So that's what I meant in the beginning, we should synergize here. Good point.
Any other question? Yes, please.
Yeah. So, Marise Borja, for those of you who don't know me already. So, Uwe, one question.
Yeah. Hi.
What's the limit of priming? Can you combine two different priming molecules and get a better effect? Can it be additive? Or really, at the end, it doesn't matter what you use to prime, you end up to a priming limit?
So we addressed this once. Once we mixed salicylic acid with BABA, and we have seen the effect was a little bit better as when we use BABA alone or when we use salicylic alone, but it was not twice as good or not even synergistic. Yeah, so it helped a little bit, but I mean, when you got them primed, they are primed, so it doesn't really help. The only thing that helps is if you prime them and it lasts for a couple of days and you prime them in this phase again, you extend the period of priming enormously, which is very important for application. So you can do a booster like in the COVID vaccination, and it helps. And basically, it's similar. Thank you.
Okay, one last question, please.
Andy Renz from FABio. Thank you, Uwe.
You're welcome.
I think, or we heard, it's difficult enough to identify, to use small molecules and figure out how they actually are involved in these SARs. So how do you do this with microbes? You mentioned some rhizobia that you are testing in AgPrime. So how can you find out if microbes actually trigger some systemic acquired resistance?
This is quite easy. So it's not. So what we have is root colonizing bacteria. Actually, many are in the market, and they are sold as a certain species. We checked that many species are wrongly annotated, so it's different ones. So, and you can check it easily. You use your culture, you do it in aqueous solution, you water the plant, and then you wait a couple of days, and then in the systemic tissue you can check for jasmonic acid. Then they are inducing the so-called induced systemic resistance or salicylic acid, and you can see SAR and prime.
Right. That's true, of course. But how do you understand what in the microbes triggers it?
Okay, yeah. So, well, we use cultures, we spin down the cells, we use the supernatant, and then we do the fractionation. I mean, I'm a biochemist, so we do all the different columns, and then we try to narrow it down to, if you are lucky, to one compound.
Okay.
And if you are even more lucky, we can identify the compound in a gas chromatography or something like that.
Thank you.
Thank you. Thanks again, Uwe.
Thank you.
For being with us.